Question number 1. A cosmid is a large bacterial plasmid, viral plasmid, hybrid of plasmid and phage yeast plasmid. Correct answer is option C, hybrid of plasmid and phage. Question number 2. Polymerase chain reaction can rapidly amplify DNA sequence of the size up to 10 kilobases, up to 45 kilobases, up to 100 kilobases, up to 1000 kilobases. Correct answer is option A, up to 10 kilobases. Question number 3. The DNA polymerase commonly used in polymerase chain reaction is obtained from E. coli, yeast, T. aquaticus eukaryote. Correct answer is option C, T. aquaticus. Question number 4. Base sequence of DNA can be determined by Maxim Gilbert method, Sanger's deoxy method, both A and B, none of these. Correct answer is option C, both A and B. Maxim Gilbert method, Sanger's deoxy method. Question number 5. Form a DNA-RNA hybrid. DNA can be obtained by addition of DNA, protein and ATP, helicase and ATP, DNA topoisomerase 1, alkali. Correct answer is option D, alkali. Question number 6. Optimum temperature of DNA polymerase of T. aquaticus is 30 degrees, 70 degrees, 54 degrees, 72 degrees. Correct answer is option D, 72 degree. Question number 7. In addition to take polymerase chain reaction require all the following except A. Template DNA, deoxyribonucleoside, type phosphate, primer, primase. Correct answer is option D. Primase is not required in polymerase chain reaction. Question number 8. DNA polymerase of T. aquaticus is preferred to that of E. coli in PCR because it replicates DNA more efficiently. It does not require primer. It is not denatured at the melting temperature of DNA. It doesn't cause error in replication. Correct answer is option C. It is not denatured at the melting temperature of DNA. 9. 20 cycle of PCR can amplify DNA. Correct answer is option A. Question number 10. Transgenic animals may be prepared by introducing a foreign gene into somatic cell of young animals, test an ovary of animals, a viral vector, and infecting the animal with the viral vector, fertilize egg and implanting the egg into foster moth. Correct answer is option D. Fertilize egg and implanting the egg into foster moth. Question number 11. Each artificial chromosome can be used to amplify DNA sequence of the size. Each artificial chromosome can be used to amplify DNA sequence of the size up to 10 kb, up to 45 kb, up to 100 kb, up to 1000 kb. Correct answer is option D, up to 1000 kb. Question number 12. DNA fingerprinting is based on the presence of DNA of constant number of tandem repeats, variable number of tandem repeats, non-repeated sequence of each DNA, introns in eukaryotic DNA. Correct answer is option B. Variable number of tandem repeats. 30. All of the following statement about restriction fragment length polymorphism are true except it results from mutation in restriction site. Mutation in restriction site can occur in coding or non-coding region of DNA. It is inherited in Mendelian fashion. It can be used to diagnose any genetic disease. Correct answer is option D. It can be used to diagnose any genetic disease is not true for restriction fragment length polymorphism. All three statements are true. Question number 40. Inborn error of urea cycle can cause all the following except vomiting, ataxia, renal failure, mental retardation. Correct answer is option A, vomiting. 50. Hyperammonia type 1 result from congenital absence of glutamate dehydrogenase, carbamyl phosphate synthesis, ornithine transcarbamylase. None of these. Correct answer is option C, ornithine transcarbamylase. 60. Congenital deficiency of ornithine transcarbamylase cause hyperammonia type 1, hyperammonia type 2, hyperornithinemia, citrullinemia. Correct answer is option D, citrullinemia. 70. A ketogenic amino acid among the following is leucine, serine, threonine, proline. Correct answer is option A, leucine. 18. Carbon skeleton of the following amino acid can serve as a substance for gluconeogenesis. Site cysteine, aspartate, glutamate, all of this. Correct answer is option D, all of this. Cysteine, aspartate, glutamate is 19. N-formine immunoglutamate is a metabolite of glutamate, histidine, tryptophan, methionine. Correct answer is option B, histidine. 20. Methylmalonyl CoA is a metabolite of valine, leucine, isoleucine, all of this. Correct answer is option A, valine. 21. Homogenetic acid is formed from homoserine, homocysteine, tyrosine, tryptophan. Correct answer is option C, tyrosine. 22. Maple syrup urine disease result from absence or deficiency of homogeneous oxidase phenylalanine hydroxylase branch chain amino acid transaminase, none of this. Correct answer is option C, branch chain amino acid transaminase. 23. Which of the following is present as a marker in lysosomal enzyme to direct them to their destination? Glucose 6-phosphate, mannose 6-phosphate, galactose 6-phosphate, and acetyluramic acid. Correct answer is option C, galactose 6-phosphate. 24. Marfan syndrome results from a mutation in the gene coding collagen, elastin, fibrillin, keratin. Correct answer is option B, elastin. 25. All the following statements about fibronectin are true except it is a glycoprotein, it is a triple helix, it is present in extracellular matrix, it binds with integrin receptor of cell. Correct answer is option A, it is a glycoprotein. Is not true for fibronectin. It is a triple helix, it present in extracellular matrix, and it binds with integrin receptor protein. Question number 26. Fibronectin has binding site for all the following except glycoprotein, collagen, heparin, integrin receptor. Correct answer is option C. Heparin is not binding site for fibronectin, glycoprotein, collagen, and integral receptor. 27. Fibronectin is involved in cell adhesion, cell movement, both A and B, none of this. Correct answer is option B, involved in cell movement. 28. Glycoproteins are marked for destruction by removal of their oligosaccharide prosthetic group, sialic acid residue, mannose residue, and terminal amino acid. Correct answer is option D, N-terminal amino acid. 29. Glycophorin is present in cell membrane of erythrocyte, platelets, neutrophils, liver. Correct answer is option A, erythrocyte. 30. Selectin are proteins that can recognize specific carbohydrate, lipid, amino acid, nucleotides. Correct answer is option A, carbohydrate.